Hello everyone. Welcome to our Science 7 lesson on meiosis, a fascinating process that plays a crucial role in how life continues from one generation to the next. Today, we'll explore the stages of meiosis, understand why it's important, and see how it contributes to genetic diversity. Let's dive in. Our goal today is to understand that cells reproduce through two types of cell division, mitosis and meiosis. We'll focus on mitosis, which is the process that helps our bodies grow and repair themselves. By the end of this lesson, you should be able to describe how mitosis works and why it's important. So, what exactly is meiosis? Meiosis is a special type of cell division. Unlike mitosis, which creates identical cells, meiosis divides a single cell twice to produce four daughter cells. These cells are called sex cells or gametes, sperm in males and egg cells in females. What's special about them? They each carry half the genetic material of the original cell. This is super important for sexual reproduction. Let's break down the key features of meiosis. Reduction division. This means the number of chromosomes is cut in half. If a human cell starts with 46 chromosomes, each gamete ends up with just 23. Two rounds of division. Meiosis happens in two parts, meiosis I and meiosis II. Genetic variation. Through processes like crossing over and independent assortment, meiosis shuffles genetic material, making each gamete unique. Haploid gametes. The end result is four haploid cells, each with one set of chromosomes. Sexual reproduction. These gametes are essential for reproduction. When a sperm and egg unite, they form a new organism with a complete set of chromosomes. Now that we understand what meiosis is and why it's important, let's go through the stages. There are two main phases, meiosis I and meiosis II, and each has four stages: prophase, metaphase, anaphase, and telophase. Let's start with meiosis I. In prophase I, the chromosomes condense and become visible. The nuclear envelope breaks down. But here's the exciting part: crossing over happens. This is when chromosomes exchange pieces of genetic material. It's like shuffling a deck of cards, creating new combinations of genes. This is one of the reasons why siblings can look so different. Next is metaphase I. The chromosome pairs line up in the center of the cell. This is where independent assortment occurs, meaning the chromosomes line up randomly. This randomness adds even more genetic variation. In anaphase I, the chromosome pairs are pulled apart and moved to opposite ends of the cell. This separation ensures that each new cell will have half the number of chromosomes, making them haploid. Now we reach telophase I. Two new nuclei form, and the cell splits into two. Each of these new cells is haploid, but each chromosome still has two sister chromatids. We're halfway there. Time for meiosis II. In prophase II, the chromosomes condense again in both haploid cells. The nuclear envelope breaks down, preparing the cells for another round of division. This second phase is necessary to create four unique sex cells. In metaphase II, chromosomes line up single file in the center of each cell. This setup is crucial for the next step, separating the sister chromatids. During anaphase II, the sister chromatids are finally pulled apart and moved to opposite ends of the cell. This ensures that each daughter cell will have just one copy of each chromosome, still haploid. Finally, in telophase II, the cells complete their division. The cytoplasm splits, and we end up with four haploid daughter cells. Each one is genetically unique and ready to become a sperm or egg cell. Let's quickly recap the stages. Prophase. Chromosomes condense, crossing over occurs. Metaphase. Chromosomes line up. Anaphase. Chromosomes or chromatids separate. Telophase. New nuclei form and cells divide. This happens twice, once in meiosis I and again in meiosis II, resulting in four unique haploid cells. And that's meiosis. From one cell to four unique gametes, meiosis is essential for life and diversity. Thank you for listening, and I hope you now see how amazing this process is.